Good morning, ladies and gentlemen, and welcome to Arab Center, Washington. Today's topic for our webinar is the prospects for developing an effective vaccine for COVID-19. When and how. Our program today is typical like our previous programs. It will be a presentation and a short conversation with our guest presenter, and then followed by Q&A for you to interact with our speaker and present your questions. You may use, if you are on Zoom, the Q&A part of the page there at the bottom of the page to send your question. Please identify yourself, affiliation if you would like, and state your question short and sweet so we can get to it quickly and give our guest an opportunity to respond directly to you. You may also use email if you are watching us on Arab Center Washington website. Use the email part of that, and that will also appear on my screen here, and I would be more than delighted to read your question, refer to you by name, and get you an answer. By way of introduction, let me say that as we speak today, the COVID-19 pandemic is still threatening, spreading, despite frantic attempts by so many governments right now to deal with it, to even pass that first stage, if you will, by reopening and resuming what people refer to as normalcy, but frankly, nobody knows what normal is, so let's refer to it as the new normal that we might be facing down the road. The numbers this morning do not look encouraging. I just, before we started, looked at the pandemic numbers globally. The cases have exceeded already 5.1 million, with 330,000 deaths worldwide. US cases have exceeded or approaching 1.6 million as we speak, with about close to 95,000 deaths. Approximately over the past 24 hours, I would say at least 2,000 deaths have happened in this country. For those who are interested specifically in the Arab world, the numbers this morning look like they're approaching 200,000 cases Arab worldwide, with about 2,500 deaths thus far. Some countries are facing or seem to be having a large basically expansion in or addition in terms of the numbers themselves. For example, Saudi Arabia this morning reported almost 2,500 cases over the past 24 hours or so, bringing it closer to like 60,000 total. About 1,500 cases added over the past 24 hours in Qatar, and so on. So this continuing threat has clearly, as you have been aware, unleashed a frantic race for a vaccine. Scientists have expressed somewhat, I would say, cautious hope, some little bit of optimism that a safe and effective vaccine can be produced in record time. The question is, what is a record time? How quickly can we really speed up a dangerous process like that? So the debate is continuing. It's actually heating up as to whether that's doable. When some countries like the US is talking about 2 or 3 months, showing some tangible results and beginning to produce a vaccine commercially before the end of the year, and talking about even millions of shots available by January. People are again going back to the old habit of saying that we will have a treatment by January that will be available to at least 300 million if needs be to all American citizens by then. Whether that's realistic or not is really the subject kind of our discussion today. As laymen, you and I and people worldwide in general are somewhat confused by the daily flow of information, because that information clearly is a mix of fact and fiction. And it's very difficult for the non-initiated, for the non-scientist, if you will, to distinguish between the two, particularly when the issue has been confused with politics. There is so much politics injected into the science at this point, it's hard to distinguish between the two. We're all aware that the US government has declared what they call Operation Warp Speed to speed up the process, because we know that research for a vaccine of this magnitude, for a pandemic of this magnitude, will take usually years, sometimes even a decade or even more. So the administration is politically anxious, I think, to show some progress for mostly political reasons. Is this doable scientifically? So we have a lot of questions that you know, continue to be asked by the media and by scientists all over the world. And today, to help us sift through all that confusion, fact and fiction, we are very delighted to have a practitioner, a man who specializes in this topic. And Dr. Ali Fattom, as indicated in the bio that we have circulated to those of you who registered for this, is Senior Vice President of Vaccines Research and Development at BlueWillow Biologics, and has 25 years of experience in vaccine research and development. He is also Adjunct Professor at the University of Michigan Nanotechnology Institute for Medicine and Biological Sciences. He initiated his career in our area here as a vaccinologist at the National Institutes of Health in the Washington, D.C. area, where he helped develop several vaccines, including, I hope I don't butcher the terms here, pneumococcus and conjugate vaccine, and headed the effort to develop vaccine for S. aureus. He later joined Univax Biologics to pursue the development of conjugate vaccines against bacterial infection, including Staphylococci. Following the merger between Univax and North American Biologics to form Nabi Biopharmaceuticals, Dr. Fattom assumed responsibility for other research activities and served as Vice President for Research between 2003 and 2010. He has a BS degree from the Hebrew University in Jerusalem and an MS in microbiology from Tel Aviv University, and a PhD in microbial ecology from the Hebrew University in Jerusalem. From 1983 to 1986, Dr. Fattom taught at Birzeit University in the West Bank of Palestine, and he is the author of over 50 peer-reviewed publications and holds more than 10 patents in his field. So Dr. Ali, welcome to Arab Center, and we look forward to your presentation. You have about 20 to 30 minutes. I know you have some valuable slides to share with our audience, and then we'll use the rest or the balance of our time for Q&A. Welcome again, the microphone is yours.
Thank you. Thank you, Khalil. I appreciate the invitation, and it's a pleasure to talk to you and also to share some data and some information about where we at, where we're going, and how fast we are going to get there. So I'll have an introduction on the viral infections in general, and we'll talk about SARS-CoV-2 versus the old SARS, where they come from and whether they are brothers or twins and so forth. Then we'll talk about the timeline of the disease, how this disease progressed from the time zero until fatality or convalescence and recovery. And also we'll talk about the vaccines as intervention to prevent the infection. And we'll talk about the different technologies and the different companies and how they approach the vaccine development. I'll also talk about the vaccinology concerns. So when you talk about more than 100 candidate vaccines and the light speed by which people are running, then you are asking yourself, really, are there any concerns? What are these concerns, especially on the background of what we have seen so far from presenting some data from one of the companies. And then we'll talk, I'll touch upon some of our technology from the University of Michigan and BlueWillow, and what we do as a vaccine and why we think that we are unique in this field. And then I'll comment on some of the data that was presented lately, and then our target vaccines, and then we'll open it for questions and answers. So the first slide is for those who forgot or never seen a infection with viruses. So viruses are particles that have to use a living cell in order to be able to propagate and to make more. So they cannot divide and propagate by themselves. So they have to attack a cell, and then they enter the cells. All they have, these particles, is DNA or RNA, and they will get into the nucleus and they take over the cell. So the cell now enslaved for the virus and start producing materials that are component of the virus. When they have enough, many viruses will be already assembled in the cell and start to get out of the cell. And this process of infection will cause all the inflammation, will cause some of those cells to go into a programmed death and to burst. And the immune system will try to get in and try to protect against this. So this is the kind of the timelines. So the major point here is really the entrance of the virus into the cell. And to do that, the virus has to attach to the cell, and then it will invade into the cell and start occupying the cell and enslave it. Now, to the COVID-19 and the coronaviruses. Coronaviruses are not new. We have coronaviruses as a regular cold infection. But the 2002, 2003, there was the SARS-CoV, and that at that time, we thought, or we know, that the virus came from bats and through a palm civet cats, and then adapted and then jumped into humans and started the epidemic at the time, which really caused deaths and infections, and but stayed at the level of epidemic, meaning it's in some areas, not across the world. While in the COVID-19, which was started in December 2019, it's the new one which is causing the pandemic, because it started to spread very fast across the world. And they are both similar. So if you look at the structure, you look at the way they do it, where they bind to the cells. So if you look at the cell, you have these crowns. These crowns, the end of the crowns, binds to the ACE2 receptor on the cells. So they both bind to the same receptor. So they are very close to each other, almost homologous. But when you look at the relationship between these two, and we're looking at the binding site, we see that there is a 74% sequence identity. And if you look at the total genome, the total RNA, there is about 80 to 90% homology between the old SARS and the new SARS. So they are not twins, but they are either cousins or non-twin brothers. So that homology tells us that they came from the same place, but this one is more developed. And if you look at the binding of the virus to the cells, you can see that the black dots here is the new SARS, the COVID-19, and the red one is the old one. You can see, for those that don't really focus on the details, the COVID-19 binds 10 times faster and stronger to the cells compared to the old SARS. And that's because of those differences that happened at the binding site on that crown. So that change in the crown adapted the cells, adapted the virus to be able to attack the cell fast and become more fit into spreading. Now, we always ask ourselves, is this is coming from the wet market and so forth? This is the infections up to January 20th, about 460, 25 confirmed cases. And what you can see is that early on, most of the cases were in the wet market in the between December up to early January. But after January, we are seeing that many of the cases that we are diagnosing actually came person to person, which is really amazing that how a virus can just go through adapting itself, get more fitness and get more selection toward more virulence, and now is jumping from one person to one person. That's the red flag that was raised, that should have raised a lot of concern at that point of time, that we have here something new. It's not like the old SARS. It is a different one. It's more virulent and more fit to jump from one person to the other. Now, if you look, and that led to more research, and now we know that, in fact, once the virus goes through the nose, and the number of virus particles in the nose start to increase with time, and for some time to increase and then reach a level, some level. And when that level, it will go to the throat, into the lung, and there where you start the disease, and the infection really becomes more virulent, and the disease starts. And as you can see here, the virus in the nose goes down when you are really very sick. And therefore, this part is what we call some asymptomatic people, asymptomatic people that have the virus in their nose, but they are not sick. And therefore, and the ability of this virus to jump from one person to the other is really very dangerous. And therefore, tackling the problem of the colonization in the nose is very important to prevent the spread of the disease, and which is a major risk factor for people. And what we've seen also that it's very infectious, and the people at risk at this point of time was the nursing homes, older people, people with some predisposition like heart disease, diabetes, and immune system, weak immune system. Those people at risk, and those people should be taken care of, including the people who are taking care of these patients, including the healthcare takers like doctors, nurses, and nursing home personnel. Those are the people that are at high risk for infection. Now, if you look at the chronology of the disease now, is that, and the first is when person is exposed through the nose, you have virus growing in the nose for 3 to 5 days. And when it reaches the high level, it starts going down into the throat and into the lungs, and the infection started. Now it becomes symptomatic. Now this period, the yellow period here, is very important to prevent, very important to treat and prevent progression of the disease. And if the disease progresses, there are two options. One option is that causes fatality, which ranges between 0.8 to 10%, but it depends on the testing and so forth. But we believe it's about 1.5%, which is about 10 times more than the flu. And the recovery, so either of you recover or you are succumb to the infection and die. So therefore, all this is the chronology, and therefore that would define for us where we are intervening. And what we call the fatality is coming because the cells, the immune system, when it starts to fight the infection, they produce a lot of cytokines, enzymes, and hormones, and so forth, and that would make like a storm. That storm is really what take yourself into the fever and all these issues with the symptoms. And some people, if the storm is strong, you cannot deal with it. You get all the organ failures, and there that will bring to death or fatality. Or if you are able to stand this storm, you can recover. Now, some of the interventions are really in the area here where we can reduce the intensity of the storm so that the immune system and the body can overcome the storm. So what kind of interventions we have? There are the preventive transmission. That's one important thing, which is the social distancing, hand washing, hand sanitization, mask wearing, because now we know it's in the nose. You have to cover your nose and your mouth so you cannot really transmit the virus from one person to the other. And also now there is nose sanitization that we can help in killing the virus, killing bacteria in the nose and so forth. That should be helping. The other part is the intervention, the treatment. Once somebody is sick, you need to treat. And I'm not going to go into the details for this presentation. There is the antivirals, remdesivir. There are repurposing some of the drugs like the famous hydroxychloroquine and also the anti-protease and so forth. All these interventions are in the going, and these are drugs being developed. The major approach is really to protect with a vaccine. Vaccine is the best thing that can happen to human race, is production of a vaccine that can prevent the virus from the infection. And also the best thing is if you are preventing colonization and the transmission. So what is the ideal vaccine? You know, if you don't have a vaccine, what kind of vaccine you're going to look for? You have to look for a safe vaccine because you are giving it to healthy people to prevent disease. So therefore, it should be safe. It should induce virus neutralizing antibodies, and we're already hearing about the antibodies. Because those antibodies, and I'll show in a second slide how this works, it prevents infection. And then you need T-cell mediated immunity. You need to kill the cells that are infected so they are not going to provide more and more virus. And that needs cell-to-cell interaction and killing by immune cells. And I think that one of the important things is to prevent spread and pandemic and epidemic. You need to induce mucosal immunity in your nose and your lungs. So even if the virus gets to the nose, it will not be able to colonize and propagate. And if it gets to the lungs, you can really prevent it from attaching to the cells in the lungs and prevent the infection. So that's very important part that I'll be focusing on going on forward. So if you look at the antibodies, if you have antibodies, the virus that gets out of the cell now is bound to the antibodies. So the spike protein, where it binds to the cells, is covered with antibody, and therefore the virus cannot bind to the receptors on the cells. That's one mechanism. The second mechanism, you need to have the T-cell mediated immunity, which is the immune cells that they look around and see which cell is infected, and they kill it and engulf it and they clear it out so the virus cannot propagate. So these are the two mechanisms that are working on how to prevent the infection in those cells. Now, we know that from Chimp Work that infection with the virus and recovery can induce immunity that the animals will not get infected again. And here is a study showing that chimpanzees were infected, allowed to recover, and then they challenged them again, and all chimps were protected. And that's a great thing, because that tells us that infection and immunity can be induced against this virus. That's a major thing to have. And which antibody you are looking for, which immunity? The major thing is this part, which we call a receptor binding domain. On the crown here, there's area which comes with the first step of the infection. If you are able to have immunity against that, then you are able to prevent the virus from sticking to the cells and preventing from infection. So almost every vaccine that is out there is focused on this area of the spike protein or the crown that we have on the virus. Almost 100 or more than 100 companies, they are targeting this area. So we know where we're going with this vaccine and what would be our target. And if you look, what are the vaccines out there? What are the technologies approach? There are several ones. Now, you have the famous ones now, the RNA vaccine that is really the sweetheart for almost everybody, including the White House, is the RNA vaccine and the DNA vaccine, where you take your RNA, the materials, and you synthesize it outside. This is the RNA on which the protein will be translated. So you inject it to the cells takes it and start to translate it into proteins. So those are the proteins. So your body is producing the proteins, which are the vaccine candidate, and then it will process it as a vaccine and generate an immune response. The other part is viral vectors. Instead of giving the coronavirus, you take a virus which is not infectious, and you allow that virus to put the corona spike protein on it. So you are really giving a non-virulent virus that will introduce the spike protein. So there are different variation of that technologies, like replicating, and everybody has his own virus that will introduce the vaccine. The classical way that we use is just to go and say, okay, you know what, I'll take that part of the protein, I'll propagate it and produce it outside in human cells or in animal cells, and then I'll purify it and I'll give it as a vaccine, which is like we do the pertussis, like the other vaccines. It's a protein. So, and this is the classical way, subunit, and there are some vaccines in the clinical trials right now. I think you all heard probably about Novavax. They have a protein here, and whether it's a free protein or a subunit or a VLP or virus-like particles and so forth. So if you look at these three categories, you can see that we are really approaching it in different ways. Now, the RNA and the DNA, which is really, as I said, is the sweetheart of everybody these days, those are non-proven technologies. There is no DNA or RNA vaccine so far. RNA vaccine's been out for many years, and there is no vaccine that beyond phase 1 clinical trials. And even Moderna, who is really having all the fame these days, they never passed the phase 1 clinical trials. They never published data that support that this technology might have a great promise. Anyway, I'll get to that in the data later on onto these. So that's really some concerns here. For the viral vectors, again, the same thing. They can produce the virus. There is some phase 1, phase 2, but there is no proven vaccine that already licensed for here. For these classical ways, almost we have every vaccine that we have is part of this. So if you look at all these categories, you can see how many vaccine companies. You have about 25 plus viral vaccines, about 40 companies. Nucleic acids, protein-based, you have about 60, and other vaccine technologies. So we are really attacking it from different direction. Now, as for developing a vaccine, is a process. That process, as Khalil indicated in his introduction, it can take 5 to 10 years. 5 to 10 years, that's assuming that you have a success. If now many of these vaccines can reach phase 1, phase 2, and phase 3 clinical trial and fail in the phase 3. So the process goes for phase 1, which is safety and immunogenicity. Phase 2, you are applying it to specific population. You look what is the dose that you're going to use, what's the schedule, and then you go to phase 3. Usually in phase 2, you get a hint whether you are in the right direction or not. But even if you have a hint in phase 2, sometimes phase 3 is failure. So the success rate under normal conditions is no more than 10%. So out of 10 vaccine candidate, maybe 1 or 2 will be successful. Now, what under the circumstances of the pandemic, we are talking about condensing this 10 years into what? According to the White House, is 10 months, 1 year or 12 months. According to other optimistic people, 18 to 24 months. So basically, they want to take 10 years and you put it down into 2 years. That's, I'm take the more conservative, two years. And that would require GMP manufacturing, which is a very complicated process, phase 1, 2, 3 clinical trials for safety, for immunogenicity, for functionality, and for efficacy. And then you have the licensure. Then you have large-scale manufacturing for millions and hundreds of millions of people. Then you need to find a way to administer it, and syringes are a huge problem environmentally and also for public health until you get the immunity. So all of this is going to happen in 18 months instead of 5 years. Now, is it doable? Possible. Now, we'll look at that later on regarding where we at and what are the issues with going this kind of progress. Remember that major thing for the FDA is safety. And I believe that the FDA and EMA will not allow a vaccine to go forward without having enough safety data. That's, if there is any political push, I'm sure the FDA will push back. And you are counting on science really to make sure that this condensation of the timelines is not going to affect the efficacy or the safety of these vaccines. Now, what are the concerns of all of this picture? The concern that we have here is that the RNA and DNA vaccines, there is no proof for successful track record. More than 20 years, always in phase 1, we never got to phase 2, phase 3. There are no peer-reviewed publication. This is the basic thing. In order to be able to say, yes, we have it, you have to have a peer-reviewed publication about your technology, your data. There is nothing like that. Safety. What is the safety of these vaccines, of these technologies? What's the quality of the antibodies and the immunity that we are getting? What is the longevity? How long we're going to have this immunity with us? There's a poor efficacy. Another concern, which is a major in my case, is that poor efficacy of IM vaccine against respiratory infections. We don't have a successful track record with the respiratory infection. Look at the RSV. It's with us until today. We don't have a vaccine for RSV. It's respiratory syncytial virus that causes in asthma in kids. CMV, SARS, and we have a vaccine for flu and we have vaccine for pneumococcus. Now, when we're gonna look into the efficacy of the flu vaccine. Now, flu vaccine, we all get flu vaccine. Now, if you look at the efficacy of the vaccine for the last 19 years, it ranges between 19% and 60%, and actually we crossed the 50% only twice or 3 times. So basically we have a great vaccine that we all take it. It's even the best case scenario are at 50% efficacy. Are we going to apply this to the vaccine that we are developing right now for SARS that will be satisfied with 50% efficacy? I don't know where these guys are going with this. So we have issues with respiratory infection and efficacy of vaccines, even with the best vaccine that we have today. So this is where our technology from the University of Michigan and BlueWillow is coming into the game. What we are saying is that these respiratory infections have to have mucosal and local immunity in addition to the immunity in the blood. So the virus gets in, you have a first line of defense in the nose and in the lungs that the virus cannot cross. And if some will cross, then the systemic immunity will take care of it. So those are creating a first line of defense by immunizing in the nose, creating immunity in the nose and in the lungs that will tackle these infections. And we think that this is a major component of that. So the technology that we have from the University of Michigan is a nanoemulsion, nanotechnology that has a antimicrobial activity, which means if you take this technology and you mix it with the virus, it will kill a virus. Mix it with a bacteria, it will kill a bacteria. And this is where we had introduced what we call NanoBioProtect. It's last week, it's been launched, is an intranasal or antiseptic with nasal swabs. So you just put it in the nose. And also we have another property that this technology with some variation can be used as a delivery system of a vaccine into the nose to induce mucosal immunity in addition to the systemic immunity. And I'll give you an example for RSV vaccine and the flu vaccines in ferrets and in non-human primates, and showing you that if you induce mucosal immunity in the nose and in the lungs, you can prevent colonization, you can prevent infection, compared to the IM where there is breakthroughs, you're not always being able to protect. So we took RSV vaccines and immunized animals in the nose or in the injectable, like the other vaccines and everybody is taking, with adjuvant. And we look at the nasal wash. After we give the animals the challenge, we immunize them 3 times, and then we give them a challenge in the nose and in the lungs. And we look, is the virus killed? Is the virus propagating there? And what is going on? Here you see on the yellow here, orange, you see the animals that receive the vaccine intramuscularly. And here is the vaccine, the red one is the vaccine received intranasally. And you can see after 7 days, the vaccine in the nose, the virus in the nose is gone. There is no virus in the nose, while all animals in the IM vaccinated animals, they still have virus in the nose. So that tells us that the intranasal immunization is giving you protection based on making first line and second line of defense, while IM immunization is giving you only the 1 line of defense, which is the line 2. And if you look at the lungs, the same story in the lungs. So the lungs you can clear with intranasal, with the mucosal immunity. You did not clear it with the intramuscular immunization. You can prevent disease, but you cannot prevent the colonization and the shedding and the spread and transmission. And that's an important part with the virus, with the respiratory viruses. And same thing here with the flu. This is pandemic flu. And you can see animals that receive intranasal immunization, there is no virus in their nose. You give them a colossal amount of virus in the nose, and then you look for it. If you are if you are infected, if you are a placebo, non-immunized animals, you have virus in the nose for 5 days. From day 1, you are not, you are clearing the virus in the nose in your case. I'm sorry, I'm trying to finish it. Now, if you look at this data, which came from the Oxford vaccine, you know, we all heard that Oxford is going to phase 2 and planning a phase 3. This is a chimpanzee's work with their vaccine. They were given intramuscular and the animals were challenged. If you look at the nasal colonization, the blue ones are the animals that were not immunized. The red are the animals that were immunized. And you can see that you did not impact the virus in the nose. And therefore, you are really, whether you're immunized or not, you are gonna carry it and give it around. If you look at the lungs, you have the animals, 2 of the 3 animals still have in the non-immunized animals. They have the virus in their lungs, and 2 out of 4 animals, 2 out of 6 animals from the immunized animals, they still have virus in the lungs. So you did not you get no protection in the nose, you get partial protection against the virus in the lungs. And of course, you know, there was no pathology in the lungs, meaning the vaccine worked in preventing pathology in the lungs, but the virus is still there, the virus is still in the nose, and therefore the expectation that you are gonna get from this vaccine should be really put in the same you know, we'll take it with a grain of salt. Don't be that optimistic. Don't be that pessimistic about it. There is a chance, but that chance probably, and what I think it's going to be no more than what we've seen with the flu vaccine efficacy. Now we come to the great news from a couple of days ago with Moderna. Moderna had a phase 1 clinical trial with their RNA vaccine, and they had 45 people in this study. They presented data from 8 people. So all this noise is coming from 8 people from this study that they presented. And they showed that it's neutralizing antibodies. They all responded with neutralizing antibodies. There are many questions and many concerns and no answers from Moderna, from anybody. First is that what about the rest of the volunteers? You have 45. And this is the first time ever that somebody who's running a clinical trial reporting data from 8 out of 45 patients and having this bombastic news about it and no answers. You know, why 8 out of 45? You should finish your clinical trial and then report it. Anyway, neutralizing antibody on day 43, about 2 weeks after the second immunization. Is that enough? What is happening at 3 months or at 6 months? Where are we at with all of this? What levels? You know, they say, okay, we generated antibodies similar to what happened with convalescing people who recover from the disease. If you look at the people who recover from the disease, the antibody and neutralizing activity ranges between 0 and high levels. So where is your number sits on this spectrum? Are they close to the 0? Or are they close to the highest numbers? So there are no numbers there, just this statement like that. And that's not really scientific. That's not really what you expect from a company with the $29 billion capital. So then do we have, why we are rushing? Why we are rushing for just going out with 8 people? What happened to the people with the high dose? You know, is the safety issues there? What are the safeties? And then this vaccine was produced and run by the National Institute of Allergy and Infectious Disease, headed by Dr. Tony Fauci. And if you look at the history of Tony for the last 30 years, every time that there is a breakthrough, Tony is on top of everything and he goes out with a statement. And the vaccine was made there, evaluated by the NIAID, and the NIAID is silent, silent, silent. So where are you guys? You know, where you stand? And that's really, all these are questions about this technology. And this is the leading vaccine that had received a colossal amount of money from the public and from the government. And I think that we are, it's a legitimate to raise those issues when it comes to that, because it was out of the ordinary way of doing this. And finally, this is what we are planning to, our plans is to take the spike protein and have it formulated with the nanoemulsion, the nanovax, and create an intranasal vaccine and go into the, and take it to the intranasal vaccine, and to prevent colonization, our target, prevent colonization and shedding and transmission in addition to preventing the disease. We are in the preclinical stage and we are a small company. So the money, all the money went to the all big ones, but we are really hoping that this kind will take track and then we get into somewhere with this vaccine. And I think I would like to really to acknowledge my colleagues, Vera Betko, Tariq Hamouda, Doug Smith, Shamala Ganson, Susan Ciotti, BlueWillow Research Team, and a vaccine research group at the University of Michigan. Go Blue. Thank you.
Thank you, Dr. Fattom. Appreciate that. You gave us a lot of information to think about. Let's dive directly into the Q&A session. We have several questions that came in already. The first one came from Mohammed Shinawi from the Voice of America. Pfizer Pharmaceutical combine two stages, referring back to, I guess, the slide you presented, two stages of testing a new vaccine promising to be ready by the end of the year. Is that feasible?
No.
Oh,
It's a big no, because remember, first of all, there are two aspects here. One is that technology is not proven. We don't have a vaccine, RNA vaccine, that even in phase 2 clinical trial. So to assume that you will have it from the first shot, that's really too much. Secondly, Pfizer knows that to develop a vaccine, and they are a great vaccinologist at Pfizer, to develop a vaccine, it will take 5 to 10 years. Actually, they have been working for more than 15 years on the Staphylococcus vaccine. They know it's not easy. Secondly, is that the immune response, we don't know what kind of immune response you are generating. We don't know how long. So therefore, it's very hard to believe that even if you go for phase 2 right away, are you going to get efficacy? Okay, what kind of efficacy you are targeting? Is 50% efficacy is enough? What kind of safety database? Can you bring a 5,000 safety? I believe that the FDA would require at least 2,500 safety database of patients. So basically, I think that there's too much, it's too early, and I don't believe any process, biological process, will take less than 18 to 24 months.
The next question came from Saeed Arikat from Al-Quds newspaper. It says, even if a vaccine is developed by early next year, is it realistic to expect that this vaccine will be effective before summer of 2021, particularly in terms of making it available to the whole world and recruit a huge army of staff to vaccinate people? Besides, you get the issue of the cost. Who will pay for 7.5 billion vaccinations?
That's a good question. You know, I think that there is a chance, if everything goes well, there is no hiccups, and we are lucky, there would be a vaccine, an indication that the vaccine works by next summer. Number one. So the next season, probably we will not have it, but we may. Now, as for the distribution and availability to other people, I think for the first time we are seeing here the combination of governments and private or non-profit organizations and funding. So, and I think also that the WHO is a major player in this, because it is the link between the first world and the rest of the world, and therefore manufacturing is going to be happening. The WHO needs to take its role in mediating and getting the technology into the right place. The third world usually don't pay that much for vaccines, because the WHO makes sure that the manufacturers, it's manufactured in different places so that it will cost less than $1 per dose. And therefore, I would think that we are not going to have the whole world vaccinated by end of next year, but definitely we'll see, if we have a successful vaccine, we will see that it will be available for many people, let's put it this way, especially that one of the considerations for the RNA vaccines and DNA vaccines is that it can be produced in large amount fast. And as we say in Arabic, al-ajali min ash-shaitan, but the devil is always there. And therefore, you cannot expect that a combination. It's really a very magic mix. Fast manufacturing, fast clinical trial, fast. And you expect to have success when you had 5 years in many cases that did not yield any success. So I will take it a little bit with patience and with some reservation.
Dr. Fattom, is it possible to get infected again with COVID-19 once you have had it before? Or is the data, as you alluded to it earlier, is still too small to make that determination?
It's too small today to make that determination, but at least from what we know about viral infections, what we know from other experiences, is that usually if you are infected and you mount a good immune response, you are protected for life. Now, many people who are recovered might not have immunity because they are sick. So basically, this is not kind of a healthy, quote unquote, recovery where your immune system is activated and then recover from that, then you have all the memory, immune memory available. So basically, it could be that some of the people will not have it, but if you are healthy and you got infected, it is most probably for a good chance that you will be protected from a second infection.
Thank you. Will this disease remain with us? Another emailed question. With us like the flu, and will we now have to take a yearly COVID-19 shot like the flu shot?
Well, that's a good question. And in Arabic, we say كذب المنجمون ولو صدقوا. You cannot predict, what is going to happen, but at least what we think is that this virus behavior is similar to what the flu vaccine, behaves. So the behavior is very similar. Therefore, we would expect that we'll have the waves, and you will have the seasonality of the infection, and it could be that it's here to stay because it's so fit that this virus, and so resembling the characteristics of the infection with the flu and RSV, and therefore it's not gonna be something that will be here and will disappear in a year or two. Number two is that with regards to yearly vaccination, the flu changes very fast. And therefore, we have a yearly injection vaccine because the virus, it changes. Now, the difference between this virus and the flu virus is that the amount of the RNA in this virus is about twice the amount that the flu has, which means this virus has enough enzymes and enough protein that can correct the translation and transcription. So basically, if something happened to the RNA, there is enough tools for this virus to correct it. It's like you have autocorrect when you write. So the possibility to have some mutation that will translate into loss of immunity and loss of specificity that really goes a little bit below compared to the flu. So therefore, the virus is steady, is fit, it can correct itself, so the changes are not happening fast like the flu. So therefore, we could be lucky and the virus will not change, and we can have a vaccine that will cover any possible emergence of any variation, which is going to be minor.
All right, we have still quite a few questions. If you don't mind giving some brief answers to the following to try to accommodate as many as possible before our time is up. How do you explain the discrepancy between the high rate of infections of COVID-19, but a lower rate who end up developing severe symptoms?
Well, it seems that it's like the flu. If you have a good immune response, immune system, and you are able to respond to the virus, you would be able to overcome that. Now, elderly, when you get older, your immune system become or you're suffering from immunosenescence, meaning that your immune system is also getting old. Therefore, the immune response would be weaker. And therefore, with age, you are become more susceptible to infections. Your immune system is not as strong. And therefore, you are more prone for infections and for progressing to disease. When you're young, you have a very good immune response, and therefore you are able to mount a good protective immune response, good memory, and therefore you could survive that. So as you can see, about 50% of the fatalities were in nursing homes, and many of the others were really because of collapse of internal organs, and also having predisposition or predisposed to the infection, like heart disease, diabetes, and other chronic diseases. So that's really makes a big difference.
Is Moderna RNA vaccine based on one or more strains of the COVID-19 virus?
There's no more. There is one COVID-19 virus, and the RBD, or the receptor binding domain, is the same. So we are not talking about different viruses like the flu. We are talking about the same virus, and therefore the RNA, their RNA is one RNA there that they have, which is related to the RBD, the receptor binding protein. And I think they have also some other areas of the spike protein integrated there. So basically, it's really, it's universal for all the COVID-19 virus.
Sophie Ibrahim would like to know, do we know the infectious dose of the virus?
No, but if you look at the nasal carriage or nasal colonization, you say, what is the maximum we reach? We reach a maximum of 10 to the 5 PFUs, or plaque forming unit, which means that there is no specific infectious dose, because they never done it. If you look at the animals, for the chimpanzees, for instance, they were challenged with 10 to the 5 PFUs, 10 to the 5, 10 to the 6 PFUs. PFUs is plaque forming unit, number of particles, about 1 million particle goes the infection in the animal. Now, it's very hard to interpret from, extrapolate the data from the chimp to humans, because animals, they always lie. So don't believe them always.
The last question will be from Dr. Suleiman Abu Badr, who thanks you for this very informative presentation, and would like to know, why do you think the percentage of COVID-19 deaths in the Arab world, and perhaps in other countries, are much lower than, for example, here in the US? Is that biological, or what other differences factor in here?
Well, I don't have nobody really is able to answer that question, because if you look at Africa, for instance, and India, they all have hydroxychloroquine for malaria and so forth. So they're like a daily treatment. And in the Arab world, maybe we are lucky for one time that we are lucky, and but I don't think that there is a genetic issue, but this is something that people need to be researched. And I think it's prone to, for researchers in the Arab world to go out and test it and check it, because that's what happened also inside Palestine, that the numbers are low, and the numbers actually came because infection in the Israeli side that were transmitted to the Palestinian side. So basically, that's an area to be discussed and to be evaluated. And I think maybe some people have started that, because if you look at the infection, for instance, inside Israel, you can see that most of the infection happened in the Ashkenazi area. So not the Sephardic. So is there something genetic background? Is there other factors? I have no idea, but need to be researched.
Yeah, I've noticed a couple of people trying to refer to also the age distribution in certain societies, including Arab society, where 65+% are under the age of 25 as a potential factor, since the COVID-19 doesn't hit younger people as much. But at any rate, Dr. Fattom, thank you so much for joining us today, for the great information that you have shared with us. And ladies and gentlemen who attended this webinar, thank you again for joining us, and we hope that you would do that again in the future. Thank you.
Thank you. Thank you, Khalil.